ANOVA, Analysis of Variance. With an analysis of variance, ANOVA, three or more conditions or groups are compared. The analysis of variance is used to determine if the observed differences among a set of means are greater than would be expected by chance alone. The ANOVA is based on the F statistic, which is similar to T in that it is a ratio of between groups treatment effects to within group variability. The test can be applied to independent groups or repeated measures designs. In a single factor experiment, the one-way ANOVA is applied when three or more independent group means are compared. The descriptor one way indicates that the design involves one independent variable or factor with three or more levels. The ANOVA can be applied to two group comparisons, but the t-test is generally considered more efficient for that purpose. Here we have a hypothetical study of the effect of using different modalities for 10 days to gain pain-free range of motion in patients with tendinitis. Through random assignment, we create four independent groups: one to get ultrasound, a second to get ice, a third to get massage, and a fourth group to serve as a control. There are 11 subjects in each group. The independent variable, type of modality, has 4 levels. Therefore, this is a single-factor, multi-level design. The dependent variable is elbow range of motion, measured in degrees. Here is the data output of the data for the hypothetical case. The test of homogeneity is not significant, suggesting that there is no difference between variance of the 4 groups, as indicated with the black circle with the 1 inside it. Like the t-test, the F statistic is a ratio of the mean square of between groups divided by the mean square of the within groups. The F statistic in this scenario suggests that there is a significant difference among the groups. In Figure 20.2a, the grand or total variance in the sample is reflected in the distribution of scores from all 4 groups around the grand mean. In Figure 20.2b, the between groups variance is determined by the distribution of the 4 group means. The error variance reflects the variability of scores within each of the groups around the group mean. In this next example, we are looking at a two-way classification of ANOVA. In this example, researchers want to compare the effect of prolonged versus quick stretch for improving ankle range of motion against a control, factor A. At the same time, they are interested in determining if the position of the knee during stretch, flexed or extended, will affect the outcome, factor B. Instead of looking at each of these factors separately, they can be combined to examine their influence: type of stretch with 3 levels and knee position with 2 levels. Within the 3x2 framework, there are 6 treatment conditions. That is what you are looking at in Figure 20.3. The descriptor two-way indicates a two-dimensional analysis involving 2 independent variables. In this example, each variable is an independent factor, not repeated. The two-way ANOVA partitions the total variance in the set of scores into between groups and error components within groups, similar to a one-way ANOVA. The between group variance explains the independent variable effects, and the error variance accounts for all sources of variation unexplained by treatment. However, because the design incorporates 2 independent variables, the between groups component must be further partitioned to account for the separate and combined effect of each independent variable. Therefore, we can ask these 3 questions. What is the effect of variable A independent of variable B? What is the effect of variable B independent of variable A? And what is the joint effect or interaction of variable A and B? These components are called main effects and interaction effects, each explaining part of the total treatment effect. In a two-way design, the effect of each independent variable can be examined separately, essentially creating 2 single-factor experiments. These effects are called main effects, illustrated in Figure 20.4a. For example, using the preceding example, we can study the main effect of stretch, factor A, by collapsing or pooling the data for the 2 knee positions. With 10 subjects in each of the original cells, we would not obtain a mean for 20 scores at each level of stretch. These three means represent the average between groups' effect of stretch, independent of the effect of knee position. Similarly, we can collapse the levels of stretch to obtain two means for the main effect of knee position, factor B. There will be 30 scores per cell, as shown in figure 20.4b. These two means reflect the average between groups' effect of knee position, independent of type of stretch. In addition to the analysis of main effects, the factorial experiment has the added advantage of being able to look at combinations of levels of each independent variable. Statistically, they are referred to as interaction effects. Interaction is present when the effects of one variable are not constant across different levels of the second variable. That is, when various combinations of levels cause differential effects. To illustrate this concept, consider the hypothetical means given for the 6 treatment groups in figure 20.5. Each mean represents a unique combination of stretch and knee position. We can plot these means to more clearly illustrate these relationships. In figure 20.5a, we have represented range of motion, the dependent variable, along the y-axis. The 3 stretch groups are represented along the x-axis. The means for range of motion for each knee position are plotted at each level of stretch, with lines connecting the means. Note that in this example the lines are parallel, which means that the pattern of response at each knee position is consistent across all levels of stretch. We can reverse the plot as shown in figure 20.5b, with knee position on the x-axis, demonstrating a constant pattern for each level of stretch across both knee positions. These graphs are called interaction plots, in this case demonstrating a situation where there is no interaction. That is, prolonged stretch will generate the highest response under both knee conditions, and knee extension scores are higher across all levels of stretch. Now consider a different set of results for the same study given in figure 20.6. The interaction plots for the data show lines that are not parallel. That is, the pattern of the baseline variable across all levels of the second variable is not constant. For example, in figure 20.6a, the plot for knee flexion indicates little difference across levels of stretch. On the other hand, the line for knee extension shows a distinct difference for prolonged stretch. In figure 20.6b, we see that the 3 flexion measures are also fairly close, but the effect of prolonged stretch with knee extension is quite different. When lines are not parallel or when they cross, interaction is present. In this example, it is not the use of prolonged stretch alone that makes the treatment more effective. It is the combination of prolonged stretch with knee extension. Therefore, there is an interaction between the two independent variables. The ANOVA will account for this difference among the interaction means as a third component of the between-group sum of squares. This slide demonstrates the results of a two-way ANOVA data analysis. There are 3 between-group sources of variance listed, 2 main effects, and the interaction effect. Stretch and position are the main effects, and stretch x position is the interaction effect. The error term represents the unexplained variability between subjects within all combinations of stretch and knee position. When the ANOVA analysis using the F statistic determined that there is a significant difference between groups' conditions, the initial analysis does not tell you which group or condition is significantly different. Multiple comparison tests are used to compare means for each significant effect following an ANOVA. There are decisions to be made about which of the several comparison tests to use. These decisions are made based on trying to control for type 1 and type 2 errors. You may explore these tests with your capstone research project next year. Lastly, be aware that there are a few more ANOVAs available. A 3-way classification is similar in theory and types of comparisons, and a 1- and 2-way classification. There is a repeated measures ANOVA where subjects serve as their own controls and outcome measures are repeated over time. There is also an ANCOVA, or analysis of covariance, where the covariate is statistically controlled for in the analysis. so that the relationship between the independent and dependent variable is analyzed with the effect of the extraneous factor removed. There will be a small amount of class time dedicated to this PowerPoint presentation about ANOVAs. We will look at the SPSS datasheet and related literature, keeping in mind that we have our debate.